All right, let me introduce myself. I'm Maureen Donovan. I'm from the University of Iowa. My background is my original training is in pharmacy from the University of Minnesota. Yay, go Gophers, to a couple of people I've already talked to with Minnesota backgrounds, and nod to Dr. Cloyd, who was at Minnesota at the time I was there also, so I know somebody historically even from this faculty. And I did my graduate work at the University of Michigan in an area called pharmaceutics, and I have been at the University of Iowa in a faculty position since that time. My research area is looking at how mechanisms of drug absorption, how drugs really get from outside the body to inside the body to hopefully where we want them in the body. So a lot of combinations of formulation, pharmacokinetics, and a few other things have kept me challenged through the years. I've worked on oral formulations. I have an interest in what I call alternative sites of delivery, and a big focus of my research has been in nasal delivery and in nose-to-brain delivery, which I'm not going to talk about at all today. So if anybody has any interest in nose-to-brain delivery for their compounds, because a lot of you neurotherapeutics individuals have sort of embraced that potential route, feel free to talk to me. I'd be happy to share some of my insights and questions along with you about that route of administration. But what I'm going to talk about today is formulation. We have a number of people, a world filled with people who have exciting new agents that could be therapeutic drug products. Yet there's a big jump between the pile of powder or what you have in a media or some other sample matrix that contains your potential therapeutic, and putting them into some sort of new package that individuals can actually use as a drug product. Okay, and we'd like our drug products in the perfect world to be worldwide available. So there are things with climate, with accessibility and so forth, with cost certainly, that we need to keep in mind as we start thinking about how is this product actually going to be used, who's going to use it, how often, how are we going to get it to them, that all roll into formulation science. So, with that, one of the first questions that when I talk to drug discovery people in particular is, well, when do I have to start worrying about formulation? And you should start thinking about formulation when you're thinking about your new molecule. You're thinking about, okay, I've got a new molecule that I think is going to be significant in Parkinson's disease. So you're starting to think, well, most Parkinson's patients in at least early stages of their disease are likely going to be able to take oral tablets. As disease progresses, maybe not. And so how am I going to administer? What are the different ways that this might be administered? Keep thinking about that, but don't focus on that. You've got to get your drug product to a point through toxicology and so forth where it really does have a high potential of getting into a human trial. Okay, so you think about it, position yourself towards a rapid then movement into a formulation that could go into clinical trials, but you don't have to have your final formulation done even before you've selected your lead compound. So that's kind of the balance, and brings me to maybe a bit better explanation of that by the time you get to your first human clinical trial, you still don't need your final formulation. By the time you get to phase 3, you probably do. Okay, and so there's this continuum that you're thinking about formulation, you're thinking about things that might be possible while you're in your early preclinical phases, you're formulating for your tox studies, you're formulating for your animal studies already. How close those formulations are to your human formulation. You'd like them to be as close as possible, but that's not always possible. So there's a fair amount of latitude in the early discovery space about what you're going to do. And in hope, my suggestion, unless you're really already starting out knowing you're going to use an enabled technology, is, keep it as simple as possible. You're going to use IV, you're going to use IP, something like that. You're looking at solution solubility issues to make sure your dose is appropriate and can scale. You might use some solvent systems that you can't translate to human, but are relatively effective to look at your early preclinical readouts. And what I have at the bottom of the slide with the not regulated is the FDA doesn't need to know all of the details, and you don't have to have all of the manufacturing control documents for these early stage formulations. Okay, as you move further in the development phase, into the development phases, you start getting to a point where documentation becomes extremely important. Okay, and so as you get to these later stages of development, when you're actually maybe conceiving of, okay, this is going to be an oral delivery system, I'm going to make a tablet for my first human trial, or I'm going to do powder and capsule. I'll talk about that in a minute. But regardless of what you do, if you're going to put it into a human, all of the information about how it was made, where the chemical came from, its characteristics, its potential chemical residues and so forth have to be documented. And so by the time you get to this later stage of development, hopefully you've had a regulatory consultant brought onto your team so that you have the appropriate documentation, laboratory sites and so forth. You're into the world of good manufacturing practices and good laboratory practices. And if your laboratory is not used to working in those, and most academic laboratories aren't, start partnering, because you're going to have to redo experiments to meet those levels of regulatory requirement if they weren't done the first time. So as you're getting into these later stages of development, a good regulatory consultant is a good thing to have, and they will guide you about how to make sure that the work you're doing is going to be fileable with the FDA. Okay, and we heard a lot about solubility yesterday. My nod to my colleagues on the stage for sort of teeing up some of the concepts that I was going to talk about today, but we've heard a lot about solubility. And so the first part of this discussion is going to be on small drug molecules. I'm going to have some information about large drug molecules too. This is the challenge of my talk, is everything from small molecules to DNA gene therapies in 45 minutes, how to formulate them. Yeah, I don't even know how to do all of it, but we'll start with the small molecules, because there's over 50 years of really good science behind what we're doing to approach small molecule formulation. But the key is it soluble? Okay, and so and if you're going to be doing even a simple intravenous or intramuscular injection in humans for your first human clinical trial, you run into the problem of your volume limited. You can only give so much volume in a intramuscular injection. You should really only infuse a reasonable number of milliliters of volume into a human IV and so forth. So that sets your volume for your concentration. If you can't get your dose into the volume that you can actually administer, you have to start thinking about your solubility issues and whether there's some way of addressing that. So when solubility is a term that's in the eye of the beholder. I've argued that a milligram per mil of solubility isn't good enough, and I've argued that a microgram per milliliter is good enough, because it depends on the agent, the target, and the route of administration. So these are just some starter points. Remember, you're volume limited at most administration sites if you're going to use a solution dosage form. There are other things besides water to put things into solution, and some of them work very well with our drug products and potential drug products. DMSO, everybody's favorite solvent. I would suspect that there's a lot more DMSO in most of your laboratories than there is in mine, because it works great in preclinical evaluations. It's not approved at any reasonable amounts for human use. So you know you've got to get your molecule out of its DMSO happy place and into usually water as you transition to that. I'm going to start testing this in humans or potentially even in some of your larger animal models. So what I have is, sort of our popular co-solvent materials, and it maybe even popular is too broad a limitation. These are about the co-solvents that are approved in humans that you can actually use in some reasonable volume that might help you to improve solubility. So you've got ethanol to work with, propylene glycol, glycerin, and DMSO topically, and there's residues that are approved, but not very much DMSO. And so you can oftentimes, and we'll let the toxicology speaker address, but you can use some DMSO in your early animal models and so forth, but you've got to solve the DMSO issue as you move forward. Where did I pull those values from and numbers? They're not secrets among the formulation people. You don't have to call me up to ask me. You're welcome to. I'm certainly happy to talk to people about their formulations as they get started. But there are compilations by the FDA, which is going to be your regulatory body that you have to work with, about what they believe is safe and effective and how much. And so this is what's called the inactive ingredient database. If you just Google inactive ingredient database, it will come up if you're more well-versed with the FDA website. It's got a ton of information on it. It actually is turned into a very good and helpful website, so I encourage you to look at it. But there is a database that I type in dimethyl sulfoxide. It tells me what routes of administration it's been approved for, at what level, and that already tells me, well, if I don't want to do further toxicology testing just on DMSO at a different level, I usually can submit my formulation at the same route of administration with an already approved level of this material, be it DMSO or cyclodextrin or whatever else I'm potentially going to use. And the FDA has already recognized that as being a safe level for human use. Okay, so inactive ingredient database, great reference source. Why do we worry about solubility? Okay, we started to talk about it a little bit, but sort of the cardinal rules for solubility is if you need to get your drug absorbed as a small molecule, it's got to be soluble because it's got to partition. Okay, it's got to diffuse across those cell membranes, and the only things that do that are molecules. Okay, so it's got to be small enough to get across the cell membranes. And in the small molecule world, oftentimes we're looking for a small molecule receptor interaction. It's got to be in solution to interact with that receptor. A small particle of your drug is not going to be the right size or steric presentation for that receptor interaction. So you do need solubility, and we like to believe that at some point in time that the body is just this huge volume of water to dissolve my drug into. And in some cases that's sort of true, that the biggest challenge is just getting it absorbed first, and then I don't really have to worry about solubility after that, but you should still keep in mind that you need to retain solubility once you've gotten the drug absorbed. So why do we need solubility? On the other hand, we talked a little bit yesterday too about, okay, it's got to be something that we call permeable. Or in early drug discovery, you look at partition coefficients, and you either measure them or you calculate them or let software packages calculate them for you. And the reason we like partition coefficients is historically people have done these wonderful correlations between the octanol water partition coefficient and the permeation rate across, whether it was a tissue or a cell culture monolayer at the time or whatever, but we get these lovely correlations. This is a scale from like -5, which Lipinski certainly doesn't love, and up to over 5, which Lipinski doesn't love either, but materials that will permeate, and the more lipophilic they are, the higher their log P, the more likely they are to partition across a cell membrane, and that makes sense. We need something that's happy in these chain layers to diffuse through a cell membrane. So we've got water solubility, which doesn't necessarily lend itself well to good permeability, and good permeability so we can get absorbed, which means we're looking at those balances. And again, there have been a number of achievements in helping us identify the characteristics of individual compounds based on their individual solubilities and partition coefficients and so forth, how to try to get that balance right. But it's a challenge. Everybody in here who's ever dealt with, 100 potential leads and getting that funnel narrowed down and so forth is, how do I do that? And then when you finally get to your balance of activity, solubility, permeability, few other things, maybe you've looked at metabolism already and so forth, and you've got your lead, and then suddenly somebody reminds you that, okay, now you've really only got 5 milliliters to put that into in your dosage form, you need more solubility, do you have to start over? That's when your formulation people go and look in their bag of tricks, and hopefully they can help try to overcome that low solubility, because you've really narrowed the funnel down to this is the compound you really need to move forward. What do you do? So this example is a history example. It's phenytoin and phosphenotoin. Phenytoin, which has been around for 50 plus years. I don't know, any idea, Jim, how long phenytoin's really been around? Okay, even longer. So well-known compound with well-known solubility problems. It doesn't have good physical-chemical properties as far as its acid-base properties and so forth. There is a salt form available that is used for IV use or IM use. Actually, probably not for IM use. I stand corrected on that likely. It's pH 11. And I don't think anybody has ever said that they liked having that infused. Because it's pH 11, it's in propylene glycol and ethanol and water. Okay. It was a salvage case. We need to be able to give this IV. It's got horrible solubility issues. This is at least a biocompatible mixture at a pH that won't kill somebody, but is certainly uncomfortable, but it's what we've got. Okay. Lo and behold, not very long ago in its history, somebody has developed a prodrug. Prodrugs are drugs that we've added something to that active drug agent to help us deliver it, help us do a few other things potentially, and then this, what we call a promoiety, is meant to be degraded off somehow, enzymatically, simple water hydrolysis, a couple of other things, rapidly once it's in the body. And so the goal here is with phosphenotoin that alkaline phosphatase cleaves the phosphate promoiety, and then we've got spontaneous recapitulation of our phenytoin. And this all happens quite rapidly once in vivo. And we don't want it happening until we administer it, but we want it to happen quickly once it's in the body. So there's a trick. We can maybe make a prodrug. We don't change the activity of the parent agent, and our goal is to be able to make this with the proper physical-chemical properties we want for administration, and then recover the active pharmaceutical ingredient, active moiety, and have it do the activity that it was designed for. So possibilities. There are other possibilities because making prodrugs is not all of them work. They all will work as their pro moiety, but not all of them crystallize, not all of them are stable, a number of other things that are problems with nearly every drug compound that you have to evaluate. But so there's other things that we can try. We already talked a little bit about co-solvents as a possibility. Limited percentages of co-solvents are certainly an appropriate thing to look at. If you're looking at oral administration too, co-solvents are appropriate. You have to worry about whether pediatric patients are going to use this because their co-solvent tolerance is different than adults. But possibilities, prodrugs, simple pH adjustment, salt forms. We can do some modification on the crystal form of the drug itself and hope. Cyclodextrins are another formulation adjuvant that have become more popular as solubility enhancers and in appropriate amounts, and if their interactions do what they need to do with your particular drug agent, they're good agents to look at. And then we continue to hope that we can solve the solubility and permeability issues by using our pretty well-known standard how to get these materials at a slightly higher solubility for administration. The third thing that I'm going to emphasize, which we really didn't talk about much yesterday, was chemical stability. Your compound's got to be chemically stable for a long time at a broad range of temperatures, we hope. Okay, so early on in your development, worry about and invest in a chemically or a stability indicating chemical assay if you can. Even for biologics, which don't have the same kinds of chemical stability assays available that we are used to for small molecules, there are typical FDA methods that people use to demonstrate that their drug product is the same when it's administered to the patient at the end of its shelf life indicated on the bottle as it was when it was manufactured. Start using or start trying to get to those stability indicating assays early, because you don't ever want to find out in sort of your late stage development that it was actually a chemical degradant that was what was the active agent, that you've been working on actually the wrong agent all along, or that when you finally started paying attention to formulation and you did some formulation work, suddenly you lost activity. And that's because the other materials you started combining did something chemically, changed your drug, and now you've lost activity and you don't really know why, because you're not monitoring its chemical stability. And I've had a number of experiences with people in drug discovery where, unfortunately, chemical stability indicating assays are expensive to develop. And usually you don't want to, you want to do other things about the biology of your compound with that money and not worry about the chemical stability. And I've had people who've had their progress slowed down considerably when they finally had to do that stability indicating assay and nothing else could move forward until it was done.
Why are they expensive? Why is it not just like the regular biology assays?
Because, well, are you a protein person? Are you an oligonucleotide person? Are you, okay, we'll just go general. So, monoclonal, well, let's go with insulin as an example, okay? Insulin, big macromolecule, lots of amino acids, but it needs that confirmation for its activity. So if I've lopped off 25 amino acids somehow, it may not work. Certainly probably not going to work as designed. Okay, but how do I, a regular chemical or a regular biological assay may not pick that up. Depends on how you're assaying. If you're using 280, 240 or whatever the UV ratios are, not ever going to pick that up. Okay, and so what you have to do to verify your protein still has the amino acid sequence and hopefully the steric confirmations for stability are well known. I don't work with large proteins very much, so they're not as well known to me. Small molecules, you have to know what their degradation pathway is. You have to be able to monitor their degradants along with the API. And so we're moving more and more to almost exclusively LCM, SMS type assays. Small molecules, oftentimes you can still use just an LC assay, but again, the world is moving more towards LCM, SMS for most of those stability indicating assays, and if you don't have LCM, SMS in your lab, you're going to have to work with somebody who does. Yeah?
So I'm trying to understand the expensive factor. Is it just because of the cost, or is it because you really try really hard to try to detect all the potential degraded products?
All of the above. So the cost is the instrumentation and the skill level of the individuals using the instrumentation and the maintenance of that instrumentation and so forth. And usually it's qualified. Most of the labs that you would likely work with are at least under GLP certifications or standards. The materials themselves aren't not necessarily the highest cost driver, but yes, figuring out what the degradants even are. So then you're into NMR. Oftentimes, and other ways of identifying what those other peaks are, because you have to actually identify what those other peaks are, not just track them, peak B, and so forth. So that's where there's a lot of time. Usually, I've seen certainly a year worth of time to do this is not out of the land of possibilities. Less time if it's relatively simple, and it's not the only thing that has to be going on. But I'm really recommending you start thinking about that in parallel with some of your mid-stage development work. Yeah?
So does this need to be GMP GLP, or can this be done earlier in the
Your earlier work that is done is fine. It's going to have to be revalidated when you've made final decisions on moving this into humans. But redoing something sometimes might be as costly as doing it the first time. Maybe not. It depends on what you're working with and how versed in what's likely going to be expected and how much interlab variability and so forth you've actually worked through in that assay development. Okay, so my rule of thumb to everybody in early stage drug discovery is keep it simple. They're going to be, you look at the world of drug products and you see all sorts of wonderful things that are part of those formulations, and you may be either wowed by them because somebody said that this magic brew worked great, you should use it too, or you may be overwhelmed. It's a whole spectrum, but if you can, in your early stage development, keep it as simple as possible. Don't add in additional factors that you've got to then go back and figure out whether they played a role or didn't play a role in excellent data or excellent activity or no activity as you're sorting things out. So you can't always win with simple, but start there. See if you can make simple work for you. But even if you're going to be, if you're lucky enough to have good solubility, good permeability at the low volumes you need to worry about, you still have to start thinking about what's the solubility as a function of temperature. Because is your chemical stability problem with your API going to put you into refrigerated or frozen storage? And if it is, you've got to worry about the solubility at lower temperatures. If you're thinking about global access, you're going to want to think about what the temperature stability at elevated temperatures is for a bit of time also. Even with solutions, especially if you're going to be administering them by injecting them into somebody, they have to be sterile. How are you going to sterilize them? Filter sterilization is not the FDA's favorite choice. Works fine in development stages, but the FDA loves autoclaves. A lot of times that's why you start thinking about that elevated temperature. Do you have a compound that can withstand that? A lot of compounds can, but biologics can't. So what do? What are you going to do to assure sterility? Are you going to have multi-dose vials? What are the preservative compatibilities and so forth? Isotonicity, osmolality are important things. Usually we solve those by adding salt or mannitol or dextrose or something. What we hope is relatively innocuous from a material standpoint, not always, so there are choices, but this is not necessarily a make or break item in your formulation, but you have to remember it. Just injecting pure water with a little bit of drug dissolved in it is not going to be particularly biologically compatible. And then pH, and I just showed you an example. pH 11 is possible. pH 3 and 4 is possible, not recommended. I mean, we like that sweet spot of pH 7 or so. Usual excursions are 4 to 5 probably, to 7 and a little bit higher. And so if you can use pH to help you with solubility, try to stay on the slightly acidic side for biocompatibility. If you're thinking about both administering a suspension parenterally, that's a real challenge because sterilization of suspensions, especially on a laboratory scale, is tough. You can't filter sterilize them. So somehow you've had to sterilize your drug. Your drug may not be stable under the usual conditions we use to sterilize solids. Okay, so sterility becomes actually significant challenge if you couldn't overcome your solubility challenge. And then we start worrying about things like particle size of your suspension, because that tells us how fast that drug is going to dissolve from that particle once you've administered it. How much the solids load is in your suspension. You've got to be able to actually syringe it into somebody, so it can't be a paste. Usually about 20% max is the solids loading in a parenteral. So oftentimes you still can't buy yourself enough solubility and solids load to get your dose in. We use suspending agents to make sure that we don't turn that into a hard cake that again can't get through a syringe or can't get pulled back out of the vial. And osmolality and pH again are considerations. So even keeping it simple, there are some other formulation materials you might have to start thinking about using to get you to a deliverable solution or suspension. Yeah.
So this is all pre-injection. How do I make sure that it stays in the solution once I put it in my compartment in the human?
Okay. Yeah, and the question is that biological solubility, how do I know that it's still soluble? And that's where we do that leap of faith. And volume, when we talk about volumes of distribution from a pharmacokinetic standpoint, we really are thinking about interstitial fluids, intracellular fluids, and something. The body's got a lot of volume, really, and we've got usually a few molecules in the same place nearby. And the nice thing about putting them into the vascular system is we've got convective flow, moving them quickly away from one another. So that's the big sort of hope. And indeed, it works really quite well. And if you've got materials that have a tendency to agglomerate, that's where you have problems. But those are fewer, so worrying about those at this moment is probably not the best use of our time. But you do have to worry. It's a good question, but typically the belief is that there's a large enough volume of fluids at 37 degrees that are helping us with solubility that if we present it correctly, we don't have to worry about localized precipitation. Infusions, on the other hand, might be, and subcutaneous infusions for people who are thinking about subcutaneous infusion of chemotherapeutics or biologics might be a slight different answer to that. Okay, I've spent a long time talking about solubility and permeability and so forth, and eventually you get to the point of throwing up your hands because you can't do that with your compound. Either it's got great solubility, but it's got terrible permeability, or it might have reasonable permeability but terrible solubility, and you want to do something about that. And so there's this world of now what I call enabling technologies, usually in the nanoscale size. So nanoparticles and dendrimers and liposomes and lipid nanoparticles and all sorts of things that have come into the both the popular delivery literature and some use even as successful drug products. And the question is, how will any of these work? Because they don't fix the blood-brain barrier problem. We all are familiar with the blood-brain barrier problem from a small molecule partitioning standpoint. A nanoparticle is bigger, much bigger than a molecule. Angstrom sizes for small molecules. Nanoparticles, 100 nanometers or so is kind of a common general size. We can make them smaller, we can make them bigger. But that didn't solve our permeability problem by making it bigger. And usually, well, for a number of other reasons, these all require some sort of endocytic uptake. Okay, so we haven't necessarily helped ourselves. We have for a number of reasons, so these are of continued interest, but the critical issues, nanoparticles have worked reasonably well for some delivery of some agents. I don't think there's a successful CNS nanoparticle delivery system yet because the blood-brain barrier is still a problem. Okay, but as if you start thinking about, okay, yep, I'm going to use a solid lipid nanoparticle, a number of other things, what should I be thinking about with those? Again, they get complicated. We have now left my keep it simple realm and we've gotten into complicated. What material are you going to use? What's your dose? Because now you've increased your total mass load because now you've got all of your nanoparticle material too as added mass. Is it biodegradable? If not, is it at least biocompatible? Does it have a hope of ever leaving the body? My gold nanoparticles, for example, have been used for a long time. Are they ever going to leave your body? I'm not sure. I haven't seen results that have convinced me yet. So these kinds of things become big issues about longer-term exposure and toxicology. What size are you going to make? It better be a selected size, not just that's the size I got, so that's the size I used. There are reasons that certain sizes do certain things, are well positioned for some activity, not well positioned for others. How much drug can you really put into that nanoparticle? If you have a monoclonal antibody, which most people aren't thinking about nanoparticle delivery, but let's use it for an example, your monoclonal antibody is 10 plus nanometers in size anyway. Putting that into or onto a 10 nanometer nanoparticle doesn't make any sense. even putting it into or onto a 100 nanometer particle. You start questioning, like, how much of that antibody can I actually associate with my nanoparticles? So the amount of drug that you can actually attach to your particles is a question that you need to wrestle with. And then how does it get out of my particle? How fast do I need it to get it out fast? Do I want it out slowly? All sorts of other things. And then where do those particles go? And I'll talk about that in a minute. Liposomes are now sort of in the big family of nanoparticles. They've been around a lot longer than a lot of these nanoparticle strategies have been. And liposomes are essentially lipid bilayers with aqueous compartments between. They sort of look like cells, so a lot of what we learned about liposomes over the last 50 years will apply to exosomes, as people might think about exosome strategies that have become more popular, and one of the biggest problems is how much drug can you put into that? Because oftentimes you make the vessel first, and then you try to load your drug. If you try to do them at the same time, it still ends up being how much drug can you really put into that system, and they are not noted to be particularly stable for long periods of time, so then we do things like freeze dry them. How do we do that and have them reform what we wanted them to be and so forth? You need people who know what they're doing with liposomes, even though they're our sort of standard package. Here are the lipids to make a liposome, get your rotavap out, and so forth, commercially available. You have to know both what you're doing and how to characterize it to make sure that you got at the end what you thought you were getting. And then I'm not going to talk about implants, but for longer term delivery, once you're very well versed with your compound and so forth, they become rational strategies if you've got a biodegradable, oftentimes larger particle that may help you in longer term administration. So just a reminder when we're speaking about size, because I can stand up here and say nanoparticles and nano and 10 to the minus 9 and all sorts of other things, but you got to kind of get that mental picture of what you're really, size. Usual cell, on the micron size. Nanoparticles, 10 nanometers, which is about, again, oftentimes what we're thinking about an antibody itself, one antibody molecule, 10 nanometers. Viruses, we start moving into slightly larger biological materials, hundreds of nanometers to approaching a micron. So when I start talking about 100 nanometer particles, which again, is kind of a particle sweet spot, they're little and they don't have much drug payload. They got a lot of other payload to make that particle, but the payload issues, and then how is my drug going to get out of my my buckyball? How is it going to get out of my carbon nanotube in the time frame I want it to? All things you have to test. And then you have to overcome Mother Nature. Because Mother Nature, every particle that we deliver to Mother Nature is well set up for moving molecules and clearing molecules and so forth. And well set up for particles that are small enough to pass through the vascular system. So again, everything's smaller than 5 microns, please, if you're going to infuse it into the vascular system. But again, we're talking about nanoparticles. That's why we can put particle matter in. It's insoluble. They're particles. But they're small enough to move through the vascular beds. But they're small enough to be cleared the way Mother Nature set up particles to be cleared out of our bodies. So foreign macromolecules, cellular debris, a bunch of other things that we're usually clearing, not necessarily through glomerular filtration. We use the reticuloendothelial system for that. Macrophages, a bunch of other cellular-based clearance mechanisms for particles. That's where our nanoparticles go. Okay, so we get high loading of nanoparticles showing up in the liver, in the spleen, kidneys, potentially in the bone marrow, the smaller nanoparticles, and not very much in the brain. Okay, the microglia in the brain will pick up particulate matter, but it doesn't mean that we can get those across the blood-brain barrier to have them be able to do that. And that's the challenge, is not losing most of your dose to clearance and getting it to where it's supposed to go. This is a fairly recent paper looking at clearance of nanoparticles and targeting of nanoparticles in cancer therapy, because that's where most of the excitement and play in nanoparticle development has been, is in cancer chemotherapy. And one of the reasons for that is that tumors don't have an intact vascular system. And so because of their poor tightness of their endothelial barrier, our nanoparticles can sneak out into the tumor mass out of the vascular system, whereas other places like the brain, the endothelial cells are keeping those particles in the vascular system and they're circulating and then eventually getting trapped in the reticuloendothelial system. So somebody started to get quantitative about where did their total dose of nanoparticles go. And while, they could detect the nanoparticles in their tumor, and yay, it seemed like a great thing, when they looked at them, what we call mass balance, 100% of what was injected IV, less than 1% actually got to the tumor of interest. And of that less than 1%, 0.0014% actually got intracellularly into one of the tumor cells for activity. But that might be good enough. We talk about how many copy numbers of things do we need to actually now produce a protein that causes a change in the presentation of a disease state, and it's not 100%. So we don't realize we're used to accepting less than 100%, but we've been accepting less than 100% for a long time in drug getting to its target. So even these low numbers are still, there's a value in evaluating them, but the value is also know where the rest of your nanoparticles went. Can't tell you how many papers I read that, they'll tell me about what seemed like the drug load or something at their site of interest and nothing about anything else. And, if you load a lot of those particles into the liver where they sort of naturally go to, naturally get cleared, is there any effect that your liver is going to have because of that? Maybe not. Maybe your therapeutic is so targeted that it actually only acts at the target site. That's awesome. But if in cancer chemotherapy, that's not the case. In most cases, small molecule cancer chemotherapy, for example. So we're going to have cellular changes in the liver too, even if we weren't treating liver cancer. And so just keep in mind, think about mass balance. Where did all the particles go? Not just looking at did they go to the one site I wanted them to? And if you put a targeting moiety on them, you don't really fix these numbers to any significant extent. So antibody targeting moieties add to size, add to complexity, haven't actually demonstrated that you get significantly, we're not going to change this number to 50%. You may get closer to this number depending on your targeting moiety, but you still have to consider the expense, the time, and the efficiency, and the off-site effects, not necessarily off-target effects that we think about with side effect profile, but off-site potential, both actual pharmacologic effect and toxicologic effects. Okay, so I'm going to leave particles and parenteral delivery and what most people are thinking about for getting their difficult to deliver material into the body. Do a little bit about simple oral administration and how to keep it simple, and try to do a little bit about macromolecules of different types in a very quick, fast-talking rest of my presentation. So again, keep it simple when you're starting out, even with an oral system. You don't have to have the tablet developed as you're doing your final tox study in dogs. If you do, great, but if you don't, a lot of times we approach first-in-human testing in something that in the business we call powder and capsule, which is exactly what you think it is. It's like I took my pure drug, I put it into a gelatin or a HPMC capsule, and that was what got administered. Okay. Solubility, we don't care. We do, but we don't pay attention to it. Aggregation of the drug, insolubility in the gastrointestinal tract, even what Kenan was talking yesterday, why you might end up with 0 in your blood compartment because your drug didn't dissolve. And hopefully you've tested things enough to have some certainty that it's going to, but simple. Powder and capsule for those early phase 1 or pre-phase 1 studies, great. But when you start thinking, when you're getting closer to phase 2, phase 3, you better have, and by the time you get to phase 3, you better have your final formulation. Sometimes these formulations take a while to come about, especially if you need some other technologies added to them. So don't wait until you've started your first-in-human study to think about how you might be formulating this, but you probably don't need to have that final or even intermediate formulation in hand as you get going on these. Again, lots of enabling technologies in the oral delivery space. If you're not familiar with them, if you don't know how to formulate them, find somebody who does, to make sure that your approach and the selection of chemicals and the characterizations and so forth are moving you along the getting to approval pathway. These are just the kind of general categories that somebody like me thinks about when I'm making a tablet. So, or when I'm thinking about a capsule or a controlled release tablet or whatever. And a lot of these are added, this API, active pharmaceutical ingredient, and then the rest of these are all added either to help me manufacture 1 million of those a minute on high speed, because it's a worldwide blockbuster, or at least just to work. So I might need a solubility enhancer. I might need some other things that work in the gastrointestinal tract to get my drug absorbed. Tablets, different kind of manufacturing processes, so different kinds of material choices, but same kind of idea. I need some other enhancements of my materials, not just my drug. Because if I'm going to deliver 10 milligrams of drug, know that a standard tablet might weigh 500 milligrams. What's all that other stuff, and why did we bother? We bothered because my 95-year-old grandmother still needs to be able to see that tablet to take it. So we just maybe have bulked it up so that it's easy to handle. And then suspensions don't have to be sterile for oral administration, but the same issues with suspending agents and so particle size and so forth need to be addressed. And then you get to this. This is what many of you are familiar with, reading the back of the package and going, why? What is all that stuff? And why is it different between this one and this one, which kind of look the same? And there are reasons, and offline, if anybody ever wants the little lesson and what the reasons for many of those materials are, let me know. I'll be happy to walk you through it. And the same thing, totally different materials for the suspension of the same actual active pharmaceutical ingredient. So they're there to help us with manufacturing, presentation, stability. Okay, macromolecules. Totally different mindset. So if oligonucleotides, proteins, gene therapies, whatever, we stop worrying as much about solubility. A lot of these are polyanions. We don't have to worry necessarily about their solubility. And it's kind of a different thing when I'm talking about a 10 nanometer molecule. Thinking about it being soluble is kind of a different concept anyway. If anybody wants to talk to me about homogeneity of solutions and so forth, we can do that too. But it doesn't matter. We don't focus on what the solubility of that macromolecule is. Doses are low, and usually chemically, they're compatible with our water systems. We don't worry about partition coefficient in most cases. Some of the smaller oligonucleotides might have some attention paid to this. But otherwise, these are not partitioning across cell membranes. Okay? They need an endocytic process to get them into a cell. Partition coefficient doesn't help you. Okay, so you don't have to worry about sort of the cardinal things that we focus on for small molecules. What we do have to worry about is stability, physical stability and chemical stability. Physical stability is all about structure. Primary structure, well, not necessarily primary structure, secondary and tertiary structure. Chemical stability is more about primary structure. And we know a fair amount about protein degradation and the kinds of amino acid sequences that are prone to degradation and so forth. Chemists try to limit them, try to change them, modify them, so we address those oxidation and deamidation potentials in our drug development, but still have to, there are times we have to worry about them, and it's these secondary structures that we really have to worry about because they're critical for activity, and they're so easy to mess up with a formulation. So we start using co-solvents and other structures and so forth, we lose our three-dimensional structure, we lose our activity. Okay, so the way to address that is, again, keep it as simple as possible. And so I'm going to give you a few examples of drug products that are in the marketplace that have been highly successful, that, to the formulators who worked on these, they would not appreciate me saying that their formulation was simple. It wasn't. A lot of work to get to these formulations, a lot of understanding in the early protein world of how do we handle these and how do we formulate them. But when I look at the formulation and when you look at the formulation and you can identify every single word in the ingredients, that's kind of in my simple. I haven't used extraordinary excipients, I haven't presented them in an unusual way, and so forth. That's simple in my definition. And those kind of additives we need to keep our simple formulation happy and shelf stable and so forth are buffers commonly get used to adjust pH and tonicity. We use amino acids as stabilizers. They have some unique characteristics and preferential stabilizing capabilities for proteins. Some other stabilizers get used also. They may look like tonicity adjusting agents, but they're also helping stabilize the protein structure, keeping it from adsorbing to surfaces and so forth, along with low levels of surfactants to help with that. Sometimes we need preservatives. Those start messing with our structures. So if you need them, you have to be careful, but they can be compatible. Some chelators, again, for chemical stabilization, EDTA in particular shows up, and again, salts for buffering tonicity effects. And so we get to the citrate-free Humira system, which has the drug and some mannitol and a little bit of polysorbate 80, which is our surfactant to help us with adsorption, and water. And it's a great product. The technology is in the single injector and making sure that the formulation is compatible with the injector system. You know, we don't want the salts precipitating out that we're keeping our formulation stable in the plungers and then causing it not to work. So again, there's a lot of effort that goes into these, but we don't have to add extraordinary formulation additives to make it more complicated and have more things potentially go wrong. And then the Nutripin has a few more ingredients, but you know, really in the world of a few additional salts is oftentimes what we see with protein formulations and antibody formulations, and I recommend starting there and hopefully ending there. And if we want to move into oligonucleotides and DNA and so forth, we're just getting bigger in our molecular size and trying to handle them. The nice thing about oligonucleotides is they are actually quite chemically stable in most cases. You know, because we think they're, you know, your body's set up RNA to be pretty stable. You know, we send enzymes after it to break it down. We don't just wait for pH changes and so forth. Same thing with DNA. Well, our RNA, DNA-based drug products have that advantage from a chemical stability standpoint, but they're also subject to all of the enzymatic activity, which oftentimes leads us to really terrible half-lives because the body's pretty good at getting rid of things that we think are good for it, but it doesn't necessarily agree. And so some of the biggest challenges to overcome in RNA delivery is how to keep your RNA stable until it gets to your target site. And hopefully, you know, stays at your target site so you don't have to dose every 30 minutes. And lipid nanoparticles have become sort of the thing of the moment to look at. All the other particle technologies have also been looked at. And what we're using those particles for, in many cases, is just to protect the RNA or encapsulate the DNA too. But they're now serving a protective function. I've hidden my actual active inside my particle. And that's a big advantage, okay? And lipid nanoparticle formulations on first blush can look pretty simple if you get over the chemical nomenclature for the lipid itself. But usually what they're containing is a bulk-forming lipid, a cationic lipid, which condenses our anionic macromolecule active, a pegylated lipid, which helps keep the surface of our nanoparticles hydrophilic and keeps lots of proteins from adsorbing and hopefully giving it a longer circulation time, and cholesterol, which stabilizes the lipid matrix in very general terms. Again, sort of simple. And a couple of years ago, we all, even as just people living in the United States and realizing that we were waiting for a vaccine, suddenly became formulation experts. which was pretty cool. You know, I had fun talking to people now about lipid nanoparticles and the differences and so forth, and why we needed them, and what was taking so long, and why minus 90. You know, minus 90 was an answer to an unknown set of stability data. We don't know. We have, you know, all of our other research suggests not room temperature, and so it was a safety valve until we got stability data, and oftentimes we can't make stability data happen faster than real time. Okay, and so now we're not worried about minus 90. We're in many cases not even worried about minus 70. We're back into regular freezer storage levels for a lot of these vaccine agents. But starting out, pegylated lipid, cationic lipid, lipid as a bulk former, little bit of buffer, and a tonicity modifier in the dispersion vehicle. You know, took a lot to make. Took a lot of science behind getting to it being this simple, but that's the component. We look at Pfizer's, same kind of thing, different lipids, different ratios, but cationic lipid, cholesterol, the RNA, mRNA strand, a little bit of buffer, and I went back and checked the ingredient list of the current vaccine, and it looks slightly different than this. You know, each time they have added or slightly changed the mRNA in the vaccines. They've modified the formulation slightly, and again, because they're learning more about that in real time, and we're seeing the changes due to those efforts. And then we start thinking about oligonucleotide therapies, and Onpattro is an siRNA therapy that's been approved. It's a lipid nanoparticle system also. I won't belabor the point outside of here we have the RNA strand in potentially a little aqueous compartment in this system, a lot of lipid as the matrix, and then the surface is more phospholipid-like or phosphate, the head group-like, and then polyethylene glycol off the surface again to give us a more hydrophilic nature to that surface to help us with distribution and limiting protein adsorption. Yesterday, great presentation about oligonucleotides, and one of the things that was kind of mentioned, and I was listening closely for, is localized delivery of these has worked out quite well for the current products. And that's oftentimes the solution to that problem of I injected it IV and most of it went to the liver and very little, not enough of it got to my site of delivery and didn't stay there long and so forth, that you'll see potentially with particulate delivery even administered IV. So as science has improved and our abilities to make the oligonucleotides more potent is probably not the right word anymore for that, but as we have lowered the dose of the actual therapeutic agent, it's lowered the volume we've needed to put it to deliver it, which gives us access to a lot of these sites as a single site delivery, and again, has been quite successful. And again, we'll hear more about this, but I thought I would at least introduce the formulation for Spinraza, which, you know, has, and again, there are people in this room who can say a lot more about the formulation of Spinraza than I can, but salts, notably some dicationic agents, which are interesting choices and purposeful. Other salts, a buffer, and this is for intrathecal delivery. Again, intrathecal delivery, you don't want a lot of other substances because they have their own local effects. So again, the Bayer ASO, simple, outside of the people who actually had to come up with how to keep it that simple and be effective and safe and everything else. We see the same kind of thing play out with viral and non-viral vectors and DNA therapies too. We're just growing in size and changing our oligonucleotides. But when we look at an adenovirus formulation, they have standard categories of materials, but nothing excessive, nothing beyond what is really needed. So stabilizers as a surfactant, potentially a solubilizing agent. This is a unique one for this particular agent. Some buffers, water, and ethanol is usually not necessary, so I suspect that that one may have been just a residual level from going through the manufacturing steps. AAV vector doesn't have ethanol in it, for example, which is why I'm thinking that that was not necessarily an intentional ethanol add for some purpose, but the vector, or the AAV vector and its encased therapeutic agent, and then sodium chloride, sodium phosphate, and poloxamer now as the surfactant. That's it. Keep it as simple as possible. Don't start adding stuff, and make sure you understand what the materials you're selecting are maybe doing to especially the tertiary structure of your agent. And yep, we're in the world of really expensive, both the drug product itself and getting the manufacturing and shelf stability world brings us into extremely expensive agents. But you know, if we get to the point where that's once in a lifetime, that we'll leave the economists to factor that out. I've said enough of this and I've taken enough time that I'll let you guys quickly look at this outside of my last one. Think about formulation development as a continuum. Think about it early, make some strategies, make appropriate formulations for your stage, knowing that you're going to have to get more serious as you get more successful. And then bring in friends. You will need all of the details done at GMP level when you file your NDA, so get a regulatory consultant in to help you about those. And then in my slide deck, I've got 2 different sources, because I can say, you know, find somebody to help you, and I know how to find those people, but I don't have any idea how to find somebody who can help me decide which adenovirus vector to use and why. Just it's a different world that I work in. So to give you guys a little bit of initial access to maybe how to go about finding, there are a couple of trade publications basically that have resource guides and vendor guides. Start with the vendors and see if they can be of any help, or if somebody makes a suggestion. Your regulatory consultants oftentimes can help you identify vendors to help along the way too. If you happen to have expertise on your campus, great. Make friends with other people who have done these kinds of work. If you've got a college of pharmacy in particular, you likely have somebody on campus who can at least get you started on some things, and then maybe connected also. So again, thank you for your attention. I don't probably have very much time as Mike is coming up with the hook, but if we've got a couple of other quick questions, I'll try to address them.
Thank you very much. It's a very nice talk. So I got a question for the formulations of RNA or DNA. Is there universal formulations for those kind of drug, or just you have to individualize, each of them?
Yeah, it's a good question, and it applies to everything. But you've asked about DNA. In particular, but is there a universal formulation? And there's a universal starting formulation. There is not a universal ending formulation. So with DNA, my recommendation is start with phosphate buffer, maybe a little bit of polysorbate 80, like 0.01%, and that might be it to start with. And then, but what you do to refine your formulation is stability, volume issues, aggregation is a big problem. And so you end up adding materials or changing out. Maybe polysorbate isn't the best surfactant. Maybe that's why poloxamer is associated with a different different product. And so you have to get down to individual evaluation of each material. But just to get started, probably phosphate buffer or citrate buffer and a little bit of surfactant gets you a long way in your early evaluations potentially even in your animal evaluations. Same thing with powder and capsule moving into a tablet. There's sort of standard tablet formulations that everybody likes to start with. They don't really work perfectly for every single drug. They don't probably even work that well for any single drug, but it gives us a place to start.
Thank you. So just one more. So sorry. Regarding the mRNA or DNA, the big molecules. So for those, seems like the lipid nanoparticle is working well than the polymer. Is that true?
So if it's DNA associated with lipid, they could have a lipid conjugated to something that's then part of the formulation, or a polymer conjugated to something, or they're actually, it is a particle and you might not recognize it or whatever. But lipids conjugated to materials, you have to think about them because those materials forming the structure that you think it's forming compared to the structure it formed with a different material. A lot of those are adsorption type interactions, and they're not, it's material A and material B, even if they're both a viral vector, they will not necessarily act the same way. So you still have to do the characterization steps. You might consider them at first blush, but you still need to characterize that what you got is what they got when they published that work to move forward.
Thank you.
For the like the mRNA vaccines, is it known or is it possible that the lipid nanoparticles have an adjuvant role as well?
Yes, potentially. I'm not sure, the current ones in the marketplace, I'm not sure how much of an adjuvant effect those particular lipids have. The cationic lipids in particular do have that tendency, and there might be a secret sauce, secret benefit to them, but you can actually use cationic lipids that don't have an immunostimulatory effect also. But I'll answer your question with a totally different world of looking at the PLGAs, the biodegradable materials that haven't made it into the marketplace yet for RNAs. They have immunostimulatory activities also, which makes them great for vaccines, but we see those materials used for a lot of other delivery routes also. So it's that matter of what's safe, and redosing usually isn't a problem. You don't get an antibody load that suddenly is reactive, but it's certainly a concern. And yeah, you can kind of, there's the secret sauce that your formulators will try to add in sometimes too.
Okay, great. Thank you very much, Maureen. We'll move on. Dr. Cloyd.